Okay, so just as a quick reminder, what we were talking about last time was the sodium potassium pump, also known as the sodium potassium ATPase. What's the rule to help us remember? Because we need to know about sodium potassium. That's actually quite important. What's the mnemonic that helps us remember? Pumpkin, right? So I'm gonna write that up again here. So pumpkin is a very easy way, right? Because what the sodium potassium ATPase, also known as the sodium potassium pump, whoops, that's very loud, sorry about that. Is it pumps K, which is an ion, of course, in, right? So if we look at our cell, we have this pump, which is a protein, of course, right? And it's going to pump potassium in. And at the same time, we know it also pumps sodium. So it ends up pumping sodium outside. So if you look at the concentration of ions, it's actually quite different. So there's a lot more, the concentration of potassium inside a cell is much greater than the concentration of potassium outside. And it's the opposite for sodium. So I just sort of draw this in. I'm not giving the exact numbers. We don't have to remember. Oops, that's big outside. This should be a small. We don't have to know the exact numbers, but it's a lot more outside, sodium outside in terms of concentration than inside, okay? So this is incredibly important because, and it comes up over and over again. This gradient is used for a lot of things. All this, well, we'll talk about that later, but all the cells in your body are actually negative inside compared to outside. And it's partially, and that's due to this pump. And that's the topic I'm not going to go into right now because that's a big, huge topic all on its own. The fact that we have a lot of sodium outside of the cell compared to inside is actually very useful because there are some forms of active transport. Do you guys remember what active transport is? There are two types of transport, two main categories, passive, active. Passive transport is the movement of molecules down their concentration gradient, right? Remember that? So that's the equivalent of this. If I go to the top of a hill, right, and have a ball, and I just release the ball, it's going to roll down, right? I don't have to do any additional thing, right? It just does it, right? Okay. People are noisy. Maybe you should close that door. Well, okay, they've calmed down now. So okay, thank you. Thank you. Sorry. So passive transport, it's equivalent of just something moving down its concentration gradient. So each molecule is dependent on its own concentration gradient. So in other words, if we're looking at, so let's look at sodium in this case. There's a higher concentration of sodium outside compared to inside. Just by diffusion, sodium might move through a sodium channel. We could put a sodium channel in here. All your cells have sodium channels. Remember, they're like pores. And of course, it's possible for some sodium to go this way, right? Through the channel. It doesn't really have a directionality to it. But overall, there's more sodium outside. So you get a net flux of sodium from outside to inside. Okay, so that's, it seems contradictory. Why would you have a pump and then have channels? Well, that's the way your cells work. You're pumping, you're maintaining a gradient in which you have more sodium outside compared to inside, and yet you also have these channels that allow some sodium to simply move by diffusion downhill. So that's passive, right? That's passive transport. Now, sodium, let me ask you a question, because we did this last time, but I know it's confusing for students. Sodium is an ion, right? Has a charge of +1. Can sodium move through the lipid bilayer all by itself? No, right? Because the lipid bilayer has this hydrophobic core. Remember the tails of the phospholipids are pointing in. The hydrophobic substances like other hydrophobic substances. They do not like things that are hydrophilic. Ions are definitely hydrophilic because they associate very well with water because they're charged. Water's polar. Those two, they go together really well. So this would be, so you need to have a channel, which is what I've drawn here, right? So it's a protein and the sodium can go through. So what's the name of that kind of diffusion? So we have a couple names. So sometimes when we refer to substance that simply goes through the membrane directly, like oxygen or CO2, sometimes textbooks will call that simple diffusion. It's really just diffusion, but they give it the term simple diffusion because it doesn't require any special protein channels or anything like this. When you have a channel like this, we refer to this as you're making it easier for the thing to do what it wants to do. You're facilitating, right? Does that ring a bell? Right. So it's a form of facilitated transport. And then there was another form of facilitated transport that we talked about also. You generally have more glucose outside of your cell, but let's do this. Let's do the same convention where I get rid of this. I'm going to draw the word glucose, big letters outside, small letters inside, to indicate that for the most part, most of our cells, we have a higher concentration of glucose outside than inside. And then you have these special transport molecules, right? They're called either transport or carrier molecules, either way it works. And the glucose can actually sort of fit in this thing, right? Remember that? And then we have a transformation in which it flips around and the glucose ends up on the other side, right? You guys recall that? So it's a way this is supposed to be an arrow. So it's doing it, it's binding to this protein on the outside of the cell. The protein changes its shape and now the glucose sort of magically ends up inside. So there's more glucose outside than inside. So by diffusion, glucose would want to move inside, right? Want in quotes, right? Molecules don't want things, but it wants to get inside, but it can't because glucose is polar, because it has these OH groups attached to it, right? Which is the case for a lot of things like sugar and alcohol. So those are hydroxyl groups. Those are polar because of the oxygen and hydrogen, and this is bound to a carbon. Oxygen is more electronegative than carbon and hydrogen. So it'll have a partial negative charge. The hydrogen here would have a partial positive charge and so forth. So it's a polar molecule. It's 6 carbon backbones, relatively large. It can't get through. So this is a type of, these are carrier proteins or transport proteins. And this is another form of facilitated diffusion, okay? So both of these facilitate to make diffusion easier for these molecules that wouldn't be able to get across your membrane any other way, okay? All right, so that's diffusion. Let's talk about active transport. This is actually active transport. Here, what does anyone recall what fuels this pump, the sodium potassium pump? Go ahead. Yeah, it's also called the sodium potassium ATPase. That's another alternative name because what it does is you have adenosine triphosphate, and that gets hydrolyzed to ADP, adenosine diphosphate, plus inorganic phosphate. Recall what phosphate is. It's PO4. It has a negative charge. Okay. So ATP, remember, is the energy currency of the cell. So you have these pumps. You're making ATP all the time because you're breaking down things like glucose. You're making ATP, and then it's fueling this pump. So again, your brain is very active, and I think I gave this is a interesting number that 70% of the energy that your brain uses, because you have a lot of cells in your brain, neurons and so forth, is just to fuel this one pump. Okay, and there's other pumps and things like that too. There's a lot more that the brain has to do, but this one pump is so important that 70% of the energy your brain uses is just for this one pump. Okay, so that's a form of active transport. You can see why it's active, right? It's using energy. We have to, ADP is a higher energy molecule. It's more complex than ATP. And so it's using up energy when you break hydrolyze ATP to ADP and inorganic phosphate. So the picture you should have of this, it's like if you had a ball or some sort of object or whatever, and you have to push it up a hill, right? That takes energy, right? Everyone can imagine that, right? So that's active transport. What are we moving uphill? We're moving sodium uphill. Sodium is low here, and it's high here, right? So we're going contrary to the way sodium would want it to simply diffuse through this channel here that I drew here. So that's moving sodium against its concentration gradient. Okay, that's the term you see a lot. Against the concentration gradient means you're moving it uphill. And at the same time, we're doing the same thing with potassium. Potassium is moving from a low concentration outside to a high concentration inside, and we're moving it uphill also against its concentration gradient. So it's considered active transport. And there's one more form of active transport that I think I mentioned briefly, but I should mention again. It's a little bit confusing. There are forms of active transport. Let's imagine this is a cell in the wall of your small intestine. And for this cell, you're going to have a different glucose transporter. So we're going to try and move glucose into the cell. So again, it's going to bind and it's going to change its shape. And the glucose will end up, I'm not drawing this very well, but you get the picture, right? The glucose will end up inside. So this is a different form of a glucose transporter, slightly different. This one, but this time, because we're trying to take the glucose from the lumen, which is the hole that goes down the middle of your small intestine where your food is, and you want to move it into the cells, you may actually have a higher concentration of glucose because you're concentrating glucose from the food, from the small intestine into the cell, and then you're going to take that into your blood, right? You're trying to get the food out of the energy, the energy, the glucose molecules out of the food that you ate. So now we're moving glucose uphill, right? Well, your body has a way of dealing with that because we're constantly pumping sodium outside. There may be sodium in the food. So if we combine the movement of sodium into the cell down its concentration gradient from a area of high concentration to area of low concentration, that can then move glucose uphill. Okay, so sodium is moving downhill, glucose is moving uphill. That's a form of active transport. And you could say, well, where's the energy coming from? Well, you could think of this sodium gradient as being like water. Think of the one hydraulic analogy is really good. You have a lot of water, right, in a dam, and then the water goes to the dam from an area of high, it's high up to low down, and you can run a turbine, and that can do work for you, right? So that's what's happening with the sodium. The sodium's like the water. It's moving downhill. We're capturing that energy and using it to fuel the movement of glucose uphill, right? And that's what your cells in the small intestine do all the time. That's another very important form of active transport that our cells do all the time, okay? Because the whole idea here is our cells are very active. Staying alive is not an easy thing. You have to constantly moving things around, gathering energy, breaking things down, and so forth, right? So that these are some of the mechanisms that we need to stay alive. All right. So let's just do a quick review of what we talked about. We have diffusion, right? We talked about that. Molecules, an individual type of molecule will move from an area of high concentration of that molecule to an area of lower concentration. Examples, across a membrane, lipid soluble molecules and gases, CO2 and oxygen, they can move directly across the membrane. So a lipid soluble molecule might be a fatty acid. It's mostly hydrocarbon with a carboxyl group at one end. So it's mostly very nonpolar. It can move straight across your membrane without any special transporters. Then we just talked about facilitated transport. We're going to always move again, from an area of high concentration to lower concentration. We're just making it a little bit easier, but it's still just basic diffusion. How do we do it? We have channels, right? I drew a sodium channel here. We also have potassium chloride channels and other types of channels. And there's carrier protein. There are carrier proteins like this glucose transporter that's very helpful. What are some examples? Amino acids, sugars and amino acids. So facilitated transport, here's glucose, that's a sugar, but you also have some amino acids, right? So you pick up amino acids across the wall of the small intestine when you absorb food. They're relatively big-ish molecules, right? And so they have to have a transporter protein to help them move across. All right, we're going to get to these next two later, exocytosis and endocytosis. These are going to be ways of moving macromolecules. So we didn't quite get to those. So, okay, so that actually brings us to the next topic. Before I move on, any questions on what I just discussed? Because I think this is some of the hardest stuff for students to grapple with. You okay for now? Okay, you can always ask me later. All right, so large molecules. We talked about macromolecules, right? They could be something like a food molecule, like a carbohydrate, right, which is a long chain of monosaccharides with the monomer. You have a polymer making this big carbohydrate. They can be pretty big. Hormones might be relatively large. Some waste could be relatively large molecules, right? Peptides, right? It's a chain of amino acids, right? It's not a peptide is often used, a terminology we use often for a shorter chain than less, roughly 100 amino acids in length. So how do we get these things across? These macromolecules are transported in or out of the cell by the vesicle formation, which I will show a picture of that in a second. And this is a pretty energy intensive process. Energy is required to form the vesicles. Vesicle formation is called membrane assisted transport. And there's two types. There's exocytosis, which we allow substances to exit from the cell. So it's kind of convenient. Exo is like exit. Cyto, you should recognize this means cell, right? And the osis just means process. So it's the cell, it's the cellular process of something exiting the cell. Endo means, it's inside, right? So it's the opposites. It's a process where which things will enter into the cell. So here's a picture. Let's look at exocytosis first. So we talked about the lipid bilayer of your plasma membrane. Well, imagine just a smaller spherical bit of lipid bilayer. That's going to be what's called a vesicle. Now, the vesicle can contain certain things. Like, for example, this might be a cell in the salivary glands in your head that are going to generate amylase, which is an enzyme that breaks down starch. So that, if you're eating corn or potatoes or something, it'd be useful. So you want to get that enzyme out of your cells and into what's called the interstitial space, and from there it ends up in your saliva. So here's this enzyme shown in green. What can happen, this is assisted by a lot of proteins, and it takes a lot of ATP and energy and so forth. So that's not being shown here. But this vesicle can fuse, its membrane can essentially fuse with that of the plasma membrane. So essentially the plasma membrane actually gets a little bit bigger. And then whatever was inside of this vesicle, is in this sort of cavity here, but then that ends up being pushed outside of the cell, okay? So this means that at other places in the cell, the membrane has to pinch off, otherwise the plasma membrane would get bigger and bigger and bigger, right? So that's how we get stuff out of a cell, through exocytosis. Cells of particular organs are specialized to produce and export molecules. A good example of this is the pancreas. Pancreas releases two different things. Hormones, right? Like insulin is a hormone, which you may have heard of. So insulin's really important for digestion. Have people heard of insulin before? What do you use? So basically when you have a meal, these pancreatic cells will release insulin, and that helps in the uptake of glucose into cells throughout your body, right? Then we have, the pancreas also makes enzymes, and these are going to be digestive enzymes that end up in the small intestine and help us break down things like proteins and so forth. Okay, so that's a good example of a kind of cell that's constant. There's cells that are releasing things all the time through this exocytosis. We also have cells in the anterior pituitary. This is an endocrine gland that lies just below your brain. It releases lots of different hormones. One example is growth hormone. Okay, so here's the opposite process. We have this thing called endocytosis. Cells take in large substances by endocytic vesicle formation. Part of the plasma membrane invaginates to envelop the substance. So this term invagination means that there's an in-pocketing, right? That's what invagination means. And it may envelop the substance, and may envelop some fluid and things like that. So the membrane then pinches off to form an intracellular vesicle. And there are three different types of endocytosis. Let's take a look at the picture though, so we get a good visual of what this process looks like. Let's take Yeah, let's take this one here. It's this. Here's this first. This one's first one's called. It's a type of endocytosis called phagocytosis. So you have the membrane. You have something. It could be a bacterium. It could be cellular debris or something like that. Imagine this might be a white blood cell on your body or some sort of immune cell that's able to do phagocytosis. It comes up. It finds this either cellular debris or bacterium. And it invaginates around it, and eventually the membrane pinches off. So now you have a complete vesicle inside. And then other vesicles with digestive enzymes will fuse with this vesicle, breaking, killing the bacterium or breaking down the cellular debris into its constituent parts, such as amino acids if it's a protein. So that's what endocytosis looks like. So this first one we just looked at, we have phagocytosis. Okay, phago literally means eating. Cyto again is cell, and then osis is process. So it's the process of eating. Okay. So what do these cells eat in your body? Typically things like immune cells will be doing phagocytosis. They're going to be eating molecules such as viruses, bacterial cells, again, cellular debris, okay, things like that. Okay. It could be food like an amoeba. You guys know what an amoeba is? They're a protist that live in ponds, and they essentially constantly on the hunt for bacteria and things like that. So for them, that's the way they get their food. The next one is pinocytosis. This is for liquids and small particles. Generally, these vesicles are smaller, and the way this is often translated, it's like cell drinking, because it's taking in smaller little sips rather than this, that's enormous amount of food at one time. So you can see what this looks like. You notice the artist has made the vesicle a bit smaller. You're taking in some fluid, some water, as well as some solutes. Okay, and your cells are doing this all the time. Pinocytosis is very common, particularly in places like capillaries. You'll move substances across capillaries, which are very fine blood vessels in your body. This is a way of getting things like glucose across those the walls of your small capillaries in the body. Okay, the next one is receptor-mediated endocytosis. So, yeah, and then there's two pictures here. We have the artist diagram, we have an actual electron micrograph. So, okay, the way this works is that there are certain substances that your cells want to pay attention to. Typically something like a hormone, which might be a peptide, right? Sort of a chain of, say, 8 or 10 amino acids in a row. That's that's kind of a relatively large molecule, so it can't just diffuse through the membrane or something like that. But this is an alternative way from having something like a carrier protein, and it works with actually relatively large molecules that carrier proteins can't work with. So imagine that the solutes a hormone. It could be insulin. Let's use the example of insulin. That's a peptide hormone. You have receptors, which are proteins that stick out from the surface all over the cell. And when the insulin binds to that, it's then these, remember, things can actually move laterally. So these receptors can pick up the insulin, move laterally, and they'll start to aggregate in an area. And on the inside of the membrane, there's this it's protein here, this sort of orangey line. That indicates there's protein there. So that's what they mean by coated. This vesicle is coated by protein. The name of the protein is clathrin. So these are clathrin-coated vesicles. And eventually, when you have enough of this molecule that we're trying to collect, like insulin, this then pinches off, and you have a coated vesicle, and then that's processed. So insulin is a signaling molecule. It's going to tell the cell, hey, there's glucose available in the around in the interstitial fluid because we just had a meal. You should be picking up some of that glucose. Okay, so that's how some of these messenger molecules will get into cells. Okay, through this method. So it's a way of just picking up very specific molecules. The cell is actually waiting there. It's like waiting for insulin to come along. Once it gathers enough, it brings it, internalizes it, and then it acts as a signal for the cell. So this is a type receptor-mediated endocytosis. The type of pinocytosis involving pits coated with receptor proteins for specific molecules. It's very efficient and specific, and again, it involves this this kind of coating. This, if you look at this electron micrograph over here, the vesicle is very dark. You see this dark line here? That's because of all this protein that's involved. Okay, so there's two types of protein. There's the receptor protein, and then the coat protein, which makes this dark line here on the outside. All right, so we're going to talk a little bit about cell surfaces. So animal cells have two different types of cell surface features. The first is extracellular matrix. So the term matrix refers to the space outside of cells that's acellular, meaning it's not actually cells, it's not living in the sense of being cells. It contains lots of proteins. It contains all sorts of solute, sugars, ions, and things like that. And it's very important. So there's a bunch of different types of extracellular matrices that we're going to talk about in this class. So this is outside of the cells. What generates the extracellular matrix? Well, the cells themselves make extracellular matrix. So you have specific cells that are constantly making this extracellular matrix. So for example, you have cells, for example, have we mentioned collagen? That's a very strong protein that's found in your skin. If you didn't have a lot of collagen in your skin, your body would just fall apart, right? So there are cells that make collagen, which is this fibrous protein, that ends up outside of those cells, right? And then and things like that. So that's a form of matrix. It's going to be the proteins and also the fluids. That's all content together. That's going to be the extracellular matrix. It's important for keeping our cells happy and healthy. And the next thing we're going to talk about is cell junctions. So if you look at your skin, if someone were to poke you, your cells don't just disintegrate and fall off, right? I mean, they do a little bit, like they're always sloughing off a few, but in general, they kind of hold together, right? And actually, if you have like dandruff or something like that, you'll see the little flakes. What's happening is the cells are actually held together by very tight junctions. So that's why the cells aren't just falling off individually. That does happen. But sometimes you'll like scrape yourself, right? And the skin will, you probably have had a bike accident or something. You'll have a little piece of like thin skin. That's because the cells are actually being held together. That's through things like cell junctions. We're going to talk about that in a second. So both of these can connect with the cytoskeleton. What is the cytoskeleton? That's the skeleton inside of the cells and contribute to cell-to-cell communication. So let's dive into this now. Let's look at this. So the extracellular matrix, it's a meshwork of structural proteins. So let's look at this. So here, things are a little flipped around. The inside of the cell is up top here, and the outside of the cell is in the bottom. It's a little bit different than how I've usually drawn it. If you look at the space between your cells, you'll have things like collagen, a very tough fiber. Elastin is a protein. It's very elastic, as the name implies. So, for example, you'll notice your ear can move, right? It has cartilage up here. It has a very high percentage of elastic, right? So if you were to sleep with your ear twisted over like this, you're not permanently disfigured for the rest of your life, right? It'll snap right back, right? Hopefully, right? That's because there you do have collagen, which gives strength, but you also have a lot of elastic fibers in the form of this protein elastin. And that gives you this elastic ability to snap right back pretty quickly. So you have collagen, elastin, provides resilience. Fibronectins is an adhesive protein that links to the cytoskeleton. So you can see this here, these green ones here. It's indirectly linking to the cytoskeleton. Remember, this is the inside of the cell. We have things like actin filaments. Again, that's another protein. And we have this transmembrane protein. It's a type of integral protein that's going from the inside of the cell to the outside. So these, you can actually have a kind of, fibronectins connected to this protein, which is connected to the actin. So you can have these sort of, strong connections between the inside and the outside of the cell due to things like fibronectin. Now, the last one I'm going to talk about here is proteoglycans. They're actually very important. So, do you guys remember we talked about glycoproteins? Does that ring a bell? So, glycoproteins, if you imagine your cell having a bunch of proteins floating around, they're like little sugars. That's the glyco part, right? They stick out and they're used for things like cell-to-cell recognition and a host of other cellular functions. So those are mostly protein. Like the exact number depends a little bit. Let's say, but just to give you some idea, like maybe it's 80% protein, 20% carbohydrate, right? That's like a glycoprotein. Proteoglycans are the opposite. They're going to be, so the proteo means protein, glycan means carbohydrate. These are going to be more in the range, and the numbers again vary, but it may be on the range of say 80% by weight carbohydrate with a bit of protein, like 20% protein. Okay, so that's why we have two very similar names that confuse everyone, including teachers, but also mostly students. You have glycoproteins, a lot of protein, a little bit of carbohydrate sticking out from the cell. You have proteoglycans, these are a little bit the opposite. So it's mostly carbohydrate with a bit of protein. These have a very different function. They're going to attach to proteins in the extracellular matrix, or they may be free floating. What they're going to do is help resist the compression of this extracellular matrix, and they may also assist in cell signaling. So they have a variety of functions outside the cell. And you see how they're drawn here? They look almost like feathers. That's actually a pretty good drawing the artist has done because they really kind of look like this. These are these proteoglycans. So they have a feathery appearance. And what they're really doing is they're essentially carbohydrates have hydroxyl groups, right? The OH groups, so they're polar. These are very attractive to water. So they hold on to water, right? So if you want to make a sort of a semi, like a gel, essentially, right? You want to have something that's watery with a high water content, but also is not too watery, that actually is a little somewhat solid, a little bit semi-solid, right? Like a gel. That's what you need is these sort of proteoglycans because they're grabbing water and holding on to it. Here's a little bit of a close-up view of what it looks like. You have this polysaccharide, you have some protein bits, and then some carbohydrates. So they're all mixed together. The carbohydrates highly attracted to water, so they're holding on to a lot of water. And this actually helps sort of puff up and hold this extracellular matrix and give it some depth and like thickness and so forth, and also resist compression. I don't know if you guys are aware of this, but water is actually highly resistive to compression, right? That's like a physical property of water. If I take a hypodermic syringe and I put a cap on the end and I have the plunger and I try and push, I can't really do anything. It will not compress, right? Whereas if I just had air inside of a hypodermic, not a hypodermic needle, what do you call it? A syringe. All right, that's what I meant to say. You have a syringe, you just close it, put a cap on the end. If it's air in it, you can compress it by quite a bit, right? Whereas if you have water, it almost feels like steel or something. It doesn't want to compress. So that's what's happening. We're attracting water, and that's actually sort of puffing up this extracellular space, and it's resistant to compression. So that's actually important for your body, right? Give strength to your body. Okay. Let's talk about cell junction. So I mentioned earlier that cells have to connect to each other, otherwise your body would fall apart. So we have these junctions between cells. So the first type we're going to look at are what's called adhesion junctions. What you have here, and a good example of this is shown up here in the corner. You have two cells and two neighboring cells. There's a very small space between them. So it's the intercellular space. There's some fluid in there. Each cell has its own cytoskeleton. So these are going to be things like filaments of actin primarily, but also there could be microtubules and things like that we discussed earlier. This is all protein here. And then there are proteins that jet out from this cell. And on the opposite, this cell, you have essentially the mirror image. You have cytoskeletal elements, protein, and then protein sticking out. And these sort of zip together, and they're tightly bound to each other. So you could think of this as being like a little spot welding job that's been done between these two cells. So this gives a tremendous amount of strength. So you find this, for example, in your skin cells. They have these little spots where you have a little weld where the cells are stuck together. And that gives a tremendous amount of strength. But at the same time, it gives flexibility. So there are spots where you have these junctions, and then they'll place it where there won't be another one. And because these little spot welds are just occurring at certain intervals, it actually gives some flexibility. So you could sort of twist and things like that and not tear. okay? So that's adhesion junctions. The next one we're going to look at is what's called tight junctions. So when you imagine you've had a meal and you want to absorb the meal across your small intestine. Well, you want to very specifically process the food molecules, right? So this is the lumen of the intestine. And what you have are cells that line. The lumen is the hole in the middle, right? Lumen. You have these cells that line this, and of course there's cells over here, right? So what you want to happen is you want to process the food and bring it in very specifically across the membrane of these cells, sort of on your terms, right? You want to say, okay, I'm bringing in certain things, but other things maybe I don't want to bring in, right? So that's something that has to happen very specifically. You don't want food particles, including things like bacteria and stuff, to just make their way between two cells, right? That would be a disaster. Now you have a bacterial infection, which is called sepsis, and you're going to die, right? So you don't want that to happen. You don't want toxins and things like that that you don't want to absorb to come into your body, right? That would be bad. So how do you prevent that? Well, you have these things called tight junctions. So the tight junctions are essentially protein that look like this. So they tie these cells together. Now you have to think a little bit three-dimensionally. If you look down on the cell, these tight junctions will go like a band all the way around the cell because it's now adjoining. There's cells all around, right? Can you imagine that three-dimensional? So they're not just in one little spot. They're not like the spot welding with the adhesion junctions. These are actually bands that go all around the cells. So the cells are completely tied to their neighboring cells. That's a tight junction, and it's shown here in electron micrograph. You can see there's a lot of protein involved in it. There's protein. There may be, in fact, multiple places where you have these junctions, right? Okay, so that's a tight junction. It's proteins that connect plasma membranes of adjacent cells. They form impermeable barriers. So this prevents what's called paracellular movement, okay? Para means around, so it prevents the paracellular movement of molecules. So the only way these molecules can get in is through your cell membranes, which is what you want. Tight junctions are found in other places in your brain. That's something called the blood-brain barrier, because again, you don't want bacteria getting into your brain. That would be a bad thing, right? What would happen? You could die, right? Your brain's very sensitive. It's very important. So you have a blood-brain barrier because of tight junctions. All the capillaries, all the blood vessels within your brain have a tight junction. They do not allow bacteria to get in, right? And that's actually, from a medical point of view, a little bit problematic. I mean, that's a good thing, but it also makes some difficult to apply to get some medicines into your brain. So they have to specifically design medicines that can go get around this problem. All right, so then gap junction. So the next type of junction we're going to talk about is what's called the gap junction. Okay, so are people okay with the tight junction? You're more or less okay. We're going to come back. These things will come up over and over again. So we're going to come back to the tight junctions and these adhesion junctions. The last one we're going to talk about here is called the gap junction. So I want to talk about your heart for a second right now. So when your heart, you're aware that your heart beats, right? So it's muscular, it beats. Okay, has anyone seen, these films are so old now, but have you ever seen a film where it could be, it's a kind of classic scene where someone the one I'm thinking of is a killer robot from the Terminator series. Arnold Schwarzenegger, he reaches his hand into this guy's chest, pulls the heart out, and it's still beating. Some people, it's like very old film, right? So the heart's beating all by itself, right? So that actually is medically correct. Your heart beats without any innervation from the brain. Now, you do have nerves that are on the heart, and they can speed up and slow down the heartbeat. But even if you cut all those nerves, which is what Arnold Schwarzenegger did and formed this killer robot from the future, the heart will beat anyway, right? It's just going to keep beating and beating, at least for a little while. So it's electrically active. There's electrical communication between the cells because they have to coordinate to do that, right? So the question then is, how are the heart cells of this first guy continuing to beat? Well, they must be connected somehow, right? So let me draw them realistically. So your heart cells look kind of like this. They're a little bit this. They have a nucleus. They're a little bit branched, okay? This is how we want to draw it. So they're all connected like this. Now, if we zoom in on this site of connection, we're going to find different types of junctions. So first of all, we're going to find the adhesion junctions, right? Which I talked about. I'm not going to draw that for now, right? But we have those because that mechanically ties these things together really well. But we have another one that I'm going to draw now, which is the gap junction. So the gap junction is essentially protein. I'll use a different color. They're actually proteins from both cells. They kind of connect up, right? So they span this intercellular space between the heart cells. Oops, I'm not drawing this very well. But they make a little channel in which water can flow between the two cells, okay? So that's a gap junction. Maybe should probably label this. These things are called gap junctions. All right, so water can pass through, small molecules like glucose can pass through, ATP can pass through, and very importantly, sodium, potassium chloride, even calcium, things like that can pass through these little passageways. Why does that have to do with Arnold Schwarzenegger, in this part? Well, electrical signals are passed via ions. So as sodium passes from one cell to another, one cell that's excited can excite its neighboring cells, and those cells become excited, and they excite their neighboring cells and so forth, right? So waves of movement of ions and so forth and electrical activity pass through the heart all the time. It's happening to you right now, right? Through these things called gap junctions. So gap junctions are incredibly important for that. The movement of things like glucose and ATP, that's also important. That's because that also provides a way, particularly for things like neurons, or sometimes gap junctions and things like that. And there are a few other types of cells where they have to pass nutrients from one another. So that's another reason we have gap junctions in our body. Okay, so small molecules move between cells. Gap junctions are too small. They will not allow things like proteins to pass through, right? Macromolecules, they're simply too big. They can't pass through. All right, so let's, any questions on that? All right, we're going to do a bunch of questions on Top Hat. Because I know there's a lot of material in that. This is, I think this is one of the more challenging in some ways chapters. Chapter 4. Okay. So you guys remember how to do this? You can go to tophat.com if you have a computer. If you don't have it, this isn't for credit. It's not a big deal, right? But you can also do it on your phone if you have an account set up. There's a join code, which is given here. Continuing to show the join code up here. I'm not really sure how the text would work. I've never done that. I think the better way is to not waste your text messages, but to set it up with an account. And again, it's a free account, right? All right, so should be able to answer now. In the fluid mosaic model, the fluid property is associated with the nature of the blank, and the mosaic pattern is established by the embedded blank. Okay, let's go ahead and take a look. People responded. So we got a little bit of a disagreement here, but most people are saying phospholipids and proteins. So when you think of the plasma membrane, You want to think of this, sometimes it's referred to as almost kind of like a sea of phospholipids. And these phospholipids form this bilayer. Again, the polar heads face out. So this would typically, the way I'm setting this up, this is the outside of the cell, this is the inside of the cell over here. So there's water inside and outside. The polar heads will look like that. The hydrocarbon tails don't like it because they're hydrophobic, so they're oriented towards the inside of the membrane. Now these little phospholipids can move laterally. They're constantly moving back and forth. There's nothing really holding them in any place. And actually, occasionally they'll even flip. Like one that's over here, it suddenly flip over to the other side. It doesn't happen often, but it does happen, right? If there's enough thermal energy, they kick from one side to the other even. So that's kind of the fluid part of this fluid mosaic model. And a mosaic is essentially a picture that's made often very beautiful in ancient times where they put tiles into a kind of cement. So this would be sort of the cement. And then the proteins would be sort of like the tiles. They're actually forming the pattern, portrait of a person made out of these colored bits of tile, okay? So the proteins sit in here. And so you can have a protein in here. This is, for example, an integral protein because it's inside of the membrane. And if it's not tied to any cytoskeletal elements or anything in the extracellular matrix, via fibronectin, which we just talked about, then this thing, even though it's pretty big, will actually move laterally, move all around. It won't flip. It's too big to do that, but it's just going to move around. This might be a receptor or something. Maybe this is it's a protein that's a receptor. It has a little part bit here where a molecule could come in and bind to it or something like that. Perhaps it can move around. Now, the other possibility is if you tie it down with things like actin filaments, like cytoskeletal elements, then it won't move around anymore, okay? But a lot of proteins do move around. It's kind of like they're like ships bobbing around in the sea. Okay, so most of you guys got it correct here. How do we go to the next thing? Yep, that's correct. Okay. All right. Let's go to the next one. Which of the following is not a function of proteins present in the plasma membrane? Okay. All right, time's up, guys. Let everyone's answered. I'll give it another 2 seconds. All right, let's see how people did. All right, so again, a little bit of disagreement. That's good. So let's look at the various things that we've got here. So proteins assist in the passage of materials into cells. Yes, right? Channel proteins, right? And also these carrier proteins, also called transport proteins, right? We talked about that last time and today also. Proteins interact with and recognize other cells. They actually do that. But when we say that, we're really talking about, remember, there's some proteins that are sitting there, and then they're going to have, I'm not really good at drawing, but this is supposed to be a carbohydrate chain that's perhaps branched, right? So what is that? That's a glycoprotein. Glycoprotein. Glyco refers to carbohydrate. So those are involved in things like cell recognition. Remember we talked about this? The immune system has to know what cells to kill, what cells not to kill. But also your neighboring cells have to recognize each other. So proteins are definitely involved in that. Proteins bind with specific hormones. Actually, I had a picture of that just a second ago, right? You could have had The way that's drawn to visualize that is you imagine there's a little notch of something or some sort right here on the top of the protein, and some sort of molecule. It could be a peptide hormone. Maybe it's made out of 8 amino acids or chained. It binds there, and then that's going to activate this protein. It may do something within the cell. What's the next one? Proteins carry out specific metabolic reactions. So you actually have that. So let's use the same example. So we have a peptide hormone that binds to our protein on the outside, but this thing, it's an enzyme on this side. So when it's activated, it may convert, it could be like converting ATP into cyclic AMP or something like that, right? It'll convert one thing to another, right? So it can do, let me draw, but there's a lot of variation in that. So let's do let me do a little picture. It's going to convert this molecule, right, into a slightly modified version of that molecule. Okay, so that's kind of a metabolic thing, right? It's a chemical change. I think that's something we're going to go into examples of that in great detail later, probably. But it does do metabolic reactions. So the one thing it doesn't do is produce lipids. Okay, so what part of your cell produces lipids? Does anyone remember? Smooth. Someone's got it. I heard something. Someone say smooth, right? No? You guys are very quiet. What? Smooth. ER, yeah, smooth ER, right? Yeah. Maybe it's my hearing. Oh, push correct. Okay, there we go. Okay, when a cell is placed in a hypotonic solution, you can talk, by the way, talk to your neighbor if you're confused. All right, we're almost out of time. All right, so everyone's got an answering. That's very good. Let's see how people did. Oh, it's all over the place. Okay, so. All right, so what I like to do, and you have, when you have an exam, and we have an exam coming up actually. So next week, we have day of service, right? You guys know that about that? And then, so we don't have a class on 29th, right? Is that a Tuesday? Oh, that's a Thursday. Okay, so on the Tuesday, all right, yeah, the 27th, we're gonna do a review, okay, review session. You have day of service on the Thursday, and then the following week on a Tuesday, which is October 4th, you have exam 1 on chapters 1 through 4, okay? All right, so it's coming up, right? Oops, what is this? So what I like to do personally, and I can bring scratch paper if you want, but you can often use also the corner of the test itself, the overwritten test, is to draw a picture, right? It'll help clarify things. So we placed our red blood cell. That's the typical way they always show it. Could be another kind of cell, but they always use red blood cells in textbooks. Put it in a hypotonic solution. Hypotonic, is it going to have more solutes? A higher concentration of solutes compared to the cell, or a lower concentration? Lower, right? Hypo means lower, right? So, so we could then draw the solutes in. And basically what we're doing is when we're talking about tonicity, I don't know if I mentioned this clearly when we talked about this. This is the sum total of all the solutes. So sodium, potassium, chloride, proteins, all the things that are soluble in there, right? So hypo means, okay, this solution really per unit volume doesn't have a lot of solutes, right? It could be maybe a little bit of sucrose or whatever it is, right? So we want to know kind of what happens to water, right? All the responses essentially looking at water. Well, actually, the first one, A, let's tackle A. Solute exits the cell to equalize the concentration of both sides. We're actually going to assume, and I told you this, right? One of the big assumptions here that we're making is that our solutes are what's called, the technical term is non-penetrating, okay? That's the technical term they use in medical textbooks and so forth. It's a solute that will not cross through the membrane. There are penetrating solutes. We're not going to deal with those in this class, okay? Because that's something I do in a more advanced class, okay? But for now, that's kind of out, okay? That's a possibility, but for what the sort of situation the solutes we're looking at, they're not going to be penetrating, they're not going to cross the membrane. And that's in fact the case for almost all solutes, okay? So it's not like a lie, it's for most of them. So now the rest of these have to do with the movement of water, okay? So I've drawn our picture, right? Is the concentration of water higher outside of the cell or inside of the cell? Outside, because solutes are physical things, they take up space, right? So there's less water molecules in per unit volume inside than outside. So this means that water will actually go both ways. There's aquaporin channels, remember those channels that allow water to go through? So some water will come out just by random chance as they bump into the aquaporin channel, will go through. But overall, you're going to have more water going in through the aquaporin channels. And so what's going to happen to our cell? It's going to increase in volume, right? So water enters the cell, moving toward the area of higher solute concentration. So I purposely left this. So this is the way lots of textbooks talk about it, and it's also a way of thinking about it. The way I like to think about it, because it's what's physically happening, is water is just like any other molecule in the universe, right? Or any other solute. It moves down its concentration gradient from area of high concentration to low concentration, right? But the way this thing is actually, the questions actually get, because you probably would have all got it right there, right? That would be too easy. But here, they sort of flip things around. They're talking about the solute concentration. Solutes are the things that are dissolved in the water. So it gives you an extra step to think about. Well, water will always move because water is just moving down its own concentration gradient, but as a consequence, it'll move into an area of high general solute concentration, right? All those solutes, protein, glucose, sodium, potassium, chloride, things like that, right? Things you'd find into a cell. So that's what's happening here. Right, so that's the correct answer. And I know this is hard, but right? But because why is it moving? Why is there more water outside compared to inside? Oh, it's because there's a higher total solute concentration inside the cell compared to outside, right? So so you see pictures of this in the textbook. Over time, what would happen is the cell would puff up, right? You get very fatter and fatter and fatter, and eventually it may lyse. Lyse means to break apart, so it may just, pop, sort of like a balloon. Okay. Okay. Again, you can discuss with your neighbors. In fact, I encourage it. You can draw a picture. I encourage drawing pictures. People are very resistant to drawing little silly diagrams. I do all sorts of diagrams, stick figures, things like that to help me understand this stuff. All right. So let's place the hypertonic solution. Wait a few more seconds. Okay, we're out of time. Let's see how people did. Okay, so again, a lot of disagreement. I kind of expect that because students have a lot of trouble with this stuff, so it's not easy. Cells placed in a hypertonic solution. So again, I drew my picture, made a red blood cell. Now this time, the solution is hypertonic. These are my solutes and little particles, right? And it doesn't matter what the nature of the particle is exactly. It could be an ion, it could be a protein, whatever. Sure. Outside than inside. So now let's think about the water. Looking at my picture, where is the water concentration higher, inside or outside of the cell? It's inside. Where I couldn't. Obviously I'd have to. I didn't continue this drawing with all the dots, right? Because it would take me an hour. But there's more water because there's fewer solids inside than outside. So water will move in which direction then? To the outside, right? So I'll draw a fat arrow this way. I might draw a skinny arrow indicating that, hey, water doesn't actually know anything, it's just kind of randomly moving. So some water will go this way, not knowing that, it's just a molecule, right? But in general, you're going to have more going out through those aquaporin channels. And so what's the correct answer here then? The water exits the cell moving toward the area of lower, water exits the cell moving toward the area of lower solute concentration. The other one is C, water exits the cell moving toward the area of higher solute concentration. Which one do you guys like? So you like B or C? C, yeah, C is the correct one, right? Because you're moving towards the area of higher solute concentration, right? Again, I purposely did this in a slightly confusing manner because if I talked about water concentration, you'd all get it right. You wouldn't have struggled at all. But this is actually a good way of thinking about it, right? Okay, let's go ahead and show the response. So that's correct. Here's an example. Has anyone ever, no one cooks, does anyone cook? Does anyone like to cook at all? Some people a little bit, yeah, okay. I like to cook, okay. So one technique for making really quick pickles is you just take some little mini cucumbers, like the Persian ones, you slice them up really quickly, throw them in a bowl, and you just put a little salt on them, right? So now I've essentially replicated this with cucumbers, which have a lot of water in them. And then I have the salt on the outside, and immediately water starts to come out. And you have this sort of salty, briny solution on the outside. And within about 20 minutes, actually quite a bit of water will come out, all this pickle juice, right? So that's essentially what's happening. The water is moving essentially from an area of high water concentration to an area of low water concentration. But you can think of those cucumbers that literally take like 20 minutes to make, which is that the water in the cucumber will move towards the salt, right? It's moving towards an area of higher solute concentration outside of the cucumber compared, of course, there's solutes inside the cell, right? There's all sorts of carbohydrates, proteins, ions, and so forth inside the cell, but there's not quite as much as when you put the salt directly on the outside. So that's why it's actually useful to think about it this way also, right? Thinking about it in terms of where are the higher solutes? Well, the water will, as the water moves down its concentration gradient, it's going to actually be moving its own concentration gradient. It will actually be moving towards an area of higher solute concentration. All right. Which of the following is incorrect regarding facilitated diffusion? Again, I encourage you guys to talk. You can think and then talk to your neighbor, see what they answered, see if you agree, argue it out if you want. Still got about 20 seconds left here. Oh, by the way, the pickles, you have to add some vinegar. I always use rice wine vinegar. Add some of that at the end. It's really good. All right, so we're down here. Let's take a look here at the responses. Yeah, so we kind of imagine they'd be all over the place. Okay, let's look at these in order. Which of the following is incorrect regarding facilitated diffusion? It's a passive process. No, that's actually correct, right? Remember, diffusion is molecules moving from an area of high one particular molecule moving down its own concentration gradient from an area of high concentration of that molecule to an area of lower concentration of that molecule. That's diffusion. So facilitated diffusion is simply just diffusion, but you've made it easier. Facilitate to make is to make something easier. You've made it easier for something like glucose to get across the membrane, or for something like an ion like sodium to get across the membrane. Okay, but it's ultimately just diffusion, right? So it's a passive process. B, it allows the movement of molecules from an area of low concentration to an area of high concentration. That's actually correct. Right? So most people got that, okay? Let's look at D. Some people answered that. It may allow for the transport of glucose across the membrane. No, that's true, right? We have glucose transporters, right? That use they move glucose by facilitated diffusion. So these carrier transporters, proteins in the membranes of all your cells actually move glucose across your cells, into your cells. All right, let's go on. Let's go on. Okay, a few seconds left. All right, see how people did. Look at the responses. Okay, so I think most people got it. So again, the sodium potassium pump, right? Okay. Pumps Kn. And that means it's an exchange pump, so it pumps sodium in the opposite direction. It's also called anti-port. Port is to carry, anti means opposite, so it's moving things in the opposite direction. So it's an exchange pump or an anti-port pump. So sodium is moving to the outside, it concentrates it on the outside. It does establish an electrochemical gradient. It's making a chemical gradient because sodium is high outside, inside, and potassium becomes high inside compared to outside. And it's an electrogenic pump, meaning it creates an electrical potential or voltage across the membrane. Does anyone remember why that would be? I didn't actually discuss this very much. Well, it was in the video we watched. It moves, there's a ratio of how many sodium for how many potassium. Does anyone remember? Say it louder.
3 potassium.
Yeah. So it's 3 sodium for every 2 potassium. So for each cycle of the pump, you're actually moving 1 positive charge outside. And that actually contributes to making the inside of the cell negative with respect to the outside. There's actually a voltage, like a battery, that's developed. So that's why it's an electrochemical gradient. That's something I didn't get it. It's something we're going to hold off until we might talk about it more later. It's a big topic. It's actually a big important topic. We didn't, but it is an electrical. So that's actually correct. Present plasma membrane. So all these answers are correct. All Which of the following processes involved in the bulk transport of molecules out of the cell? Okay, everyone's answered, so let's go ahead and look at the responses. Okay. So exocytosis is correct. So exo, think exit, cyto means cell, the O system just means process. So the cell is the process of bringing something outside of the cell. All the other ones, phagocytosis is like solid eating, pinocytosis is sort of like cell drinking. Receptor-mediated endocytosis, that's for bringing in very specific molecules like insulin, right? They bind to the receptor, they get gathered together. Then you have this protein-coated head. Protein changes flapper and it pinches off and makes something inside. Which process uses special proteins on the surface of the membrane? Proteins on the surface of the membrane to identify specific molecules for transport into the cell. Now. Let's stop it a little earlier here. 30 seconds. Okay, let's see how people did. Most people have responded. So in this case, it's receptor-mediated endocytosis. The last one, you don't have to answer if you're headed out the door. If you want to, you can answer though. I think this is going to be the last one. Gas junctions. We're kind of at the end, so do you want to just shout the answer out? What do you guys think? No one? What do gas junctions do? They're in between our egg cells. See how people do it. Oh, you're all over the place, guys. They allow for the movement of small molecules between neighboring cells, right? Okay. Remember that? Sodium, potassium chloride, ATP, glucose, water, of course. They're supposed to move through. Okay, thanks. So, yeah, so I'll see you on Tuesday. We'll have a review next, right? And then we have the day of service, and the following week we have our first exam. Okay. Thank you.